In chapters 2 and 3, we started looking at amino acids and proteins, and then we took a break and started to explore genes and genomes, DNA, RNA. But now we're coming back to proteins in chapter 7. And chapter 7 allows us to take a look at how proteins function. And we do this by looking at the example of myoglobin and hemoglobin. So here we're going to look at a protein in action. So just to outline what we're going to talk about, we will look at the structures and the functions of hemoglobin versus myoglobin, oxygen transport and storage, mechanisms of binding, that binding of oxygen there. We will look at cooperative binding and allostery, so sequential versus concerted models of binding. We'll talk about something called the Bohr effect, and then protein evolution and mutations that are present. So hemoglobin and myoglobin can be used to illustrate protein function. They're similar substances. One possesses a quaternary structure and the other only possesses a tertiary structure. So this allows us to make comparisons between these two substances. So hemoglobin and myoglobin, again, as we said, can be used to illustrate protein structure and function. They're two evolutionarily related proteins. They have nearly identical structures in terms of their binding for oxygen. There are some differences between the two. So in terms of the differences that we have, the hemoglobin is more efficient at binding O2. So that hemoglobin is more efficient, and we see that because hemoglobin can utilize 90% of its binding capacity. So the hemoglobin itself, 90% of the binding capacity. Whereas the myoglobin only uses 7% of that binding capacity. Now another difference, myoglobin is a single polypeptide chain, and hemoglobin is a tetramer with 4 polypeptide chains. So both of these structures will bind the oxygen. The hemoglobin, because there are 4 chains, each chain can bind oxygen, and it does so through cooperative binding. And the other difference is that hemoglobin, of course, we're talking about our red blood cells in our blood, and the myoglobin is going to be in muscle cells. So this is just the overall myoglobin structure. So we can see that's that single polypeptide chain, and then which is represented by these blue portions here. And then we have this group in the middle, which is a prosthetic group that we'll talk about in a little more detail here in the next slide. But that's our heme group, and that's what's actually going to help bind the oxygen. So in terms of that prosthetic heme group, this is a protoporphyrin ring. So we refer to it as a protoporphyrin ring. It does have an organic portion, which is made up of pyrrole rings, and we can see this pyrrole ring here. So we see these structures in more than one place. Got those pyrrole rings. You see that the nitrogen of the pyrrole rings, you have the 4 nitrogen atoms here. They are going to be coordinated to this iron. Okay, so iron is going to be at the center of these structures. Now iron is going to be stabilized by these nitrogens, but also it's stabilized by a proximal histidine residue. So one of the histidine residues from the overall protein chain, that polypeptide chain, is able to interact with and stabilize that iron. Now, iron can be in both the 2+ and 3+ state. So both of these can exist within the structure. However, the 2+ state is the only state that's capable of binding oxygen. So if we're trying to bind oxygen, we want the 2+ state. Now, so when we're looking at this iron within the structure, our iron in the center, iron has 6 coordination sites. Okay, so iron coordinates with each of the 4 nitrogens, so 1, 2, 3, 4. That histidine residue, that's 5. And then the 6th coordination site is where our oxygen is going to come in and bind to this structure. And so here we're looking at the binding of that oxygen. And we actually can see in this particular case, we're looking at a side view of that prosthetic group, or that protoporphyrin ring here, this heme group. And we can see our iron that's in the center of the group here. Now, within this ring structure, that's where our 4 nitrogens are going to interact with that iron. And this is actually showing us that histidine residue that comes in and helps stabilize that iron. So on the left here, this deoxyhemoglobin, this has 5 coordination sites to that iron. And then we bring oxygen in, and that oxygen is going to coordinate to that 6th site, or coordinate with that iron. Okay, so deoxyhemoglobin is missing the oxygen. Oxyhemoglobin has the oxygen. Now, when iron binds, we said that the iron that's in the 2+ state, that is the preferred state for oxygen binding. That's the only state that can bind oxygen. But we can have this 2+ state be converted, once it's bound, to a 3+ state. And when that happens, we call that a superoxide ion. So the interaction that's here, when that oxygen binds, you end up with kind of a resonance structure that occurs, and that resonance structure is between that 2+ and that 3+ state. So dioxygen and the superoxide ion. And even though this resonance structure does exist, it's important that when our oxygen is released, so the whole purpose of the hemoglobin or myoglobin is to carry oxygen to tissues. And so when that oxygen is released, we need it to be released when it is the dioxygen. We don't want these superoxide ions to be released. And the reason for that, the superoxide ions, these are very reactive. And because they're reactive, they can be very damaging to biological materials. Another reason that we don't want this superoxide ion to be released into tissues would be because if we released it in this form, then we leave the iron in the 3+ state. If the iron is left in the 3+ state, it's no longer able to bind oxygen. So no additional oxygen can bind. So we lose that oxygen storage capacity for that particular structure. And we do have a built-in mechanism of sorts to prevent the release of that superoxide ion. And the way that that happens is that the pocket for oxygen binding is really stabilized further by additional residues from that the protein chain. So what happens is, remember we said that our iron itself was going to be coordinated to, we have these nitrogens here, and then there's a histidine residue that's going to interact with this proximal histidine residue. And then this 6th binding site is that oxygen. So here's our oxygen right here. Well, this oxygen is further stabilized by another histidine residue, our distal histidine residue. So that helps stabilize that species, that bound oxygen species, so that we minimize the release of that superoxide. Now, the distal histidine, while it does aid in the stabilization of the oxygen and prevents the release of that superoxide, it has another advantage to it. It has been found that it may also limit the access of carbon monoxide to the heme. So it's going to prevent that carbon monoxide from going in there. And what happens is, if carbon monoxide can interact with that heme group and bind, it binds tightly, and that has significant consequences to it. So we looked at that myoglobin structure, which was that single chain. Now with the hemoglobin structure, we said hemoglobin has that quaternary structure here, and that arises from taking these individual polypeptide chains and through intermolecular interactions allowing them to interact with one another. Okay, so these are 4 polypeptide chains that are interacting with one another, and we see that on here. We have what we refer to as beta chains and then alpha chains. Okay. Now the alpha and beta here, this is just the notation for the chains. It doesn't mean we have beta sheets versus alpha helices. I do just want to point that out. So in this overall structure, we have these 4 chains that come together, and you'll see within each chain we have that prosthetic group, we have that heme group within the structure. What this is showing us here is the deoxyhemoglobin. So this is the structure when it's not bound. So this is unbound oxygen here. Now both the myoglobin and the hemoglobin are going to bind oxygen. Myoglobin, when we look at the binding curve of oxygen, myoglobin's curve has a sharp rise and then it levels off. And we see that here on the left. Here's our sharp rise and then a leveling off of the curve. So that is what happens with myoglobin. And the half saturation point is at a low pressure for the myoglobin. So about 2 torr. So what that means is, again, the pressure when half of the total number of oxygen that can bind to our overall sample, when that occurs, we're at a pressure of 2 torr. So we'll talk more about what that means in a second. It does indicate that oxygen will bind myoglobin with a very high affinity. It doesn't take much pressure to get it to bind. Now for hemoglobin, the binding curve of the hemoglobin, we'll see on the right here, it has more of a sigmoidal shape to it. So here's our hemoglobin binding curve. And this is because hemoglobin binds oxygen cooperatively. So binding of one of the oxygens makes it easier for other oxygen molecules to bind. So it's a property that's exhibited in proteins that show cooperative binding behavior. Now here, the half saturation point is at a higher pressure. So the half saturation point we see is 26 torr. Okay, higher pressure. This means that oxygen binding is going to be a little weaker in the hemoglobin, or it means that it's going to take greater pressures to get that to bind. So this binding that we observe in hemoglobin versus myoglobin does have physiological significance to it. Part of this is based on where we know these different substances are. So remember that we said myoglobin was going to be in muscle, and then hemoglobin, that's in the blood, and the blood, we're filtering through the body. So at some point, this is going to interact with the lungs. So if we're looking at this overall, we know oxygen is transported from the lungs to actively metabolizing tissues. Okay, well, in the lungs, the pressure is about 100 torr. Okay, so here we're showing what's going on in the lungs. We've got that pressure of 100 torr. So we bind in the lungs, then we take it to tissue, and the pressure in the tissue, as we said, about 20 torr. So pressure of oxygen is high in the lungs and it's low in the tissue. The high pressure in the lungs allows for the binding there in the hemoglobin that's present, and the low pressure in the tissues allows for the release of oxygen at that point. So with hemoglobin, 98% of the sites are bound in the lungs. So when that's moving through, about 98% of the possible sites for oxygen binding have bound oxygen to them. As it moves through the tissue, only about 32% of the sites are going to be occupied. What that means is, as it travels from the lungs to the tissues, oxygen is released. So that means that 66% of the potential binding sites are contributing to oxygen transport. Okay, we had 98% were filled. By the time we get to the tissues, 32% are filled. That means 66% of those sites, oxygen has been released. It contributes to the overall transport of the oxygen. Now, myoglobin, it's not a substance that would be effective for oxygen transport. That's because it would have 98% of its sites bound in the lungs, but those sites would remain bound when they are in the tissues because of that half saturation point. So the myoglobin would bind the oxygen in the lungs and not want to let it go. So hemoglobin is better at transporting oxygen overall, and then the myoglobin is more useful when we're dealing with just the tissue. So here we're looking at kind of the response to exercise in terms of what happens to that oxygen saturation. The overall take home from this graph is that because in both rest and exercise, the pressure correlates with the steepest part of the curve. So hemoglobin will be effective at providing oxygen to tissue where it's needed in both cases, when we're resting and when we're exercising. Okay, so remember that the hemoglobin, this curve we know is hemoglobin because it has this sigmoidal shape to it. So we bind in the lungs, and resting and exercise, we're going to be able to release some of that oxygen. Now here we're looking at that structure of hemoglobin. We do have a conformation change when we have oxygen binding to the hemoglobin. It's a slight change, but that does change the properties. So on the left we have the deoxyhemoglobin, and on the right we have oxyhemoglobin. So the deoxy is the unbound, and the oxy is the bound form. And I think the most obvious difference will be in terms of this little pocket in the center here. But what happens is when you have that binding, you end up having one of the alpha beta dimers. So remember we said that this was a tetramer, and we have the beta and the alpha, and the beta and the alpha. But we think of these as being dimers. So this would be one dimer, and this would be the other dimer down here. So one of those alpha beta dimers will shift with respect to the other, and it rotates it about 15 degrees, and that rotation closes this pocket slightly. Now we tend to refer to this deoxyhemoglobin as being in the T state. And the T state, what we mean is the tense state. And the oxyhemoglobin we say is in the R state, and that is the relaxed state. In the R state, our oxygen binding sites are free from strain, and they are capable of binding oxygen with higher affinity than the sites that we see in the T state. So that shift in structure is pretty important in terms of the implications for binding oxygen. So we can describe the binding of the oxygen within this overall hemoglobin structure, and we have a couple of different models that allow us to do this. We have the concerted model, which we'll look at here, and then in a few slides we'll look at the sequential model. So in the concerted model, we start with the subunits in their T states. Okay, so everything is going to be in the T state, and the T state is shown with the boxes here. The R state is shown down here. So as one oxygen comes in and binds, it slightly shifts our equilibrium, but the T state is still preferred. And we see that because we're paying attention here to the equilibrium arrows. So even though when we're looking at this, we can have the T or the R state, based on the equilibrium, the T state is strongly favored. We know that because the arrow pointing towards the T state is greater. So we prefer to be in the T state. And oxygen comes in and binds, and we see this oxygen coming in to bind. And when that oxygen comes in and binds, we're still preferring the T state. It does shift the equilibrium slightly. So if you're looking at the magnitude of these two arrows, the arrow towards the R state gets a little bit bigger, but T state is still favored. And then we have a second oxygen that comes in and binds, and we see that the equilibrium is shift a little bit more. So at this point, the T and the R state become equally favored. So with increasing oxygen bound, the R state is going to become more and more favored. Once we get a third oxygen that comes in, you see that your equilibrium shifts, and now your R state is preferred. And when the fourth is there, then your R state is strongly favored. So the R state, remember, that's the fully oxygenated state. And then the deoxyhemoglobin would be in the T state. So this is just illustrating that the R state favors binding, okay, by looking at these curves. So we can look at this is the saturation, so how much is actually going to be bound, and the pressure at which it's bound. And we see that in the T state, not really a very high fractional saturation, but when we're in the R state, very high. Now, what's observed is something in between that, and we'll get to that in a second. So the second model we have to describe this binding of oxygen in hemoglobin is the sequential model. In this model, the binding of oxygen results in conformational change in a neighboring subunit, and that makes it easier for something else to come in and bind. So what happens is we have our T state, and then our oxygen comes in and binds, and when the oxygen binds, it changes the conformation of that particular portion. When that conformation changes, it makes it easier. It changes the conformation in a neighboring. So it was square over here. We had a square here, and it becomes curved here, changes that conformation. And changing that conformation makes it easier for another oxygen to come in and bind. And then when that oxygen binds, it changes the conformation of the neighboring, which makes it easier for another oxygen to come in and bind. So in the sequential, we end up having a conformational change in the subunit, which allows or induces changes in neighboring subunits, and those changes increase the binding affinity. Now we see these two models, but actually neither model in its pure form will really describe the behavior of binding. It's actually a combination of the models that we need in order to describe how the binding occurs here. But it's useful to know these two models and what their implications are. So we said there were some conformational changes that take place upon binding of the oxygen. So there's our binding of oxygen here. And so what we're looking at, the gray here is the deoxyhemoglobin, or the T state, and the red is the oxyhemoglobin, which is the R state. And so you see that slight shift in the structure when you have bound versus unbound. Now we do need some sort of regulation of the R and T state so that the proper levels of oxygen can bind and release to the hemoglobin. So we actually have a mechanism in place, and the mechanism comes in the form of this 2,3-bisphosphoglycerate, or 2,3-BPG. So this is something that's present in the cells. Without the 2,3-BPG, the hemoglobin would bind the oxygen and it wouldn't let it go. And we see that in this curve. If we didn't have this substance, this 2,3-bisphosphoglycerate, you would end up having binding, and that binding affinity, even though we're decreasing the pressures down here, is still pretty high. Okay, so it binds it more tightly and it wouldn't release it properly. Whereas when we have it present, that's where our nice sigmoidal curve comes into play. And remember, that means that it will bind tightly in the lungs but then release in the tissue. So this molecule, this 2,3-bisphosphoglycerate, is important for allowing these things to actually bind and release. So in terms of the binding, this is just showing the interaction of that 2,3-bisphosphoglycerate with the residues, and it's the residues on the beta subunits. And this process will help stabilize the T form. So this is just showing there's our 2,3-BPG, kind of in that pocket there. And these are just the particular residues of the polypeptide chains that are going to interact with that 2,3-BPG. Now, to further illustrate this role of 2,3-bisphosphoglycerate, hemoglobin, fetal hemoglobin, it doesn't bind the 2,3-bisphosphoglycerate as well as maternal hemoglobin. And this is because fetal hemoglobin has different chains. It has 2 gamma chains instead of 2 beta chains. So fetal hemoglobin is a gamma 2, it's an alpha 2, gamma 2, whereas maternal, we have that alpha 2, beta 2. Okay, so we have a slightly different structure in terms of that chain. Now, because of this, the fetal hemoglobin has a higher binding affinity for oxygen. This is going to allow oxygen to be transferred from the maternal to the fetal red blood cells. But it shows that the fetal cells are going to hold on to it more tightly. It doesn't have that strong interaction with the BPG, so it's not going to release it as easily. And we see that based on the the fractional saturations at particular pressures when we're comparing both the maternal and the fetal. Now, we do often talk about the importance of pH, physiological pH, and maintaining physiological pH. We do see an effect of changing pH on the affinity for oxygen within the hemoglobin. And so this figure here is just showing how slightly changing the pH. Here we're changing from 7.4 to 7.2. You can see how it shifts the curve, and it actually causes a significant difference in binding affinity here. So pH is very important for maintaining that binding affinity. So the binding affinity does change with pH, but it also is going to change with changing carbon dioxide concentration. And so the Bohr effect is something that is going to examine or explain how the affinity of binding of oxygen changes with carbon dioxide. So we know that carbon dioxide is a byproduct from body tissue, from different cellular processes that are occurring. And carbon dioxide will dissolve in the blood to form carbonic acid. The carbonic acid will dissociate into bicarbonate and hydrogen ions. And so that's kind of what we're seeing here. So we have our body tissue, and our carbon dioxide is going to be released from the processes, and that's going to be dissolved in the blood. And once it's dissolved in the blood, we have this combination with water, and we're forming that carbonic acid, which then will dissociate, and you'll form bicarbonate and hydrogen. Okay? When this happens, because we're forming these hydrogen ions here, we are changing the pH of the blood. We're decreasing the pH. So there will also be no oxygen present in this case because we're using this respiration. So the presence of this hydrogen, it's going to result in protonation of side chains of that hemoglobin structure. That's going to alter the binding affinity of the T state. So it alters the binding affinity. The T state will become favored upon protonation of those side chains. And it's stabilized, and so our binding affinity is altered. So by having carbon dioxide present, what this idea of this Bohr effect is that carbon dioxide being present produces protons. Those protons alter the pH. Having that pH altered is going to alter how those side chains in our overall protein structure look. By altering those, we're going to alter the binding affinity of our overall protein. Now there is a second mechanism which occurs when carbon dioxide is present. And in the second mechanism, we have a direct interaction between carbon dioxide and the hemoglobin structure. And so here we're looking at one of the residues on that polypeptide chain, and we're showing how this carbon dioxide can interact with that to form this carbamate. And we're also producing that hydrogen there. So again, this would help stabilize the T state and result in the release of oxygen from our structure. We can further examine this by looking at this figure here. So the presence of carbon dioxide decreases, just in general, decreases the affinity of hemoglobin for oxygen. So this is allowing us to compare both just pH changes where we have no carbon dioxide present, and then what happens where if we're at the same pH and carbon dioxide is present, meaning that it's that second mechanism where the carbon dioxide is interacting directly, how this alters the overall binding. And we see that the binding affinity is going to be decreased, and we see that because that fractional saturation is going to decrease as we're moving from our 7.4 pH to our 7.2 to our 7.2 with carbon dioxide. Now the ultimate goal is to take this carbon dioxide that's produced in tissues and be able to exhale it. We want to get it out of the body. And so we have these two mechanisms in place. Again, in the first, we're converting it to that bicarbonate. So typically, most of the carbon dioxide is transported to the lungs in the form of that bicarbonate that's produced in red blood cells. And then it can be released in the blood plasma and travel and then be excreted. A lesser amount will be transported by the hemoglobin in that form of that attached carbamate. Okay. So we just want to get it from the tissues out so that it can be carried in the red blood cells, it's carried, and then it's released to the lungs where we can exhale it. Now, just to show the importance of that amino acid sequence and our overall protein structure, sickle cell anemia is a blood disorder of hemoglobin. About 7% of the world's population has some sort of disorder that it's a disorder of hemoglobin, and it's caused by some variation in the amino acid sequence. So remember, if we vary the amino acid sequence, we can alter the overall structure, and altering the structure is going to alter the function. So sickle cell anemia is an example where we've had a change in that amino acid sequence. So that change in the amino acid sequence in sickle cell anemia, we actually have a single amino acid residue that's going to be substituted in that beta chain, and that's responsible for it. So what we have is we have the replacement of a glutamate residue with a valine, and that's in position 6. So in normal hemoglobin, this is going to be glutamate. Here it's valine that's substituted in. And so when we have this mutation, we refer to this as hemoglobin S, rather than the normal chain, which we would call hemoglobin B. So when this mutation takes place, it actually is going to decrease the solubility of deoxyhemoglobin. So we end up with, because it's decreased solubility, we end up with aggregates of this forming. So those aggregates are forming based on what's going on in the different portions here. So here we're looking at oxygenated versus deoxygenated. And in this figure, the red triangles are representing that mutation that can occur to form that hemoglobin, that S hemoglobin. Now, we will end up with, in our deoxy, in both cases, we end up with these little hydrophobic patches that are formed based on the arrangement of the amino acids that are present. So when we have the altered hemoglobin, or that hemoglobin S, when it's in its deoxygenated form, it actually becomes complementary and can interact with one another so that these mutations fit nicely into of a neighboring deoxy, these little hydrophobic pockets there, and causes our aggregates to form overall. Now this figure is just showing that we do have a rather high occurrence of sickle cell anemia in individuals in West Africa, and part of that, so you'd wonder why would we have that? This is such a devastating disease, but part of that is because there's a correlation between areas where malaria is high. So there's a correlation between regions with high frequency of that mutation in our hemoglobin and regions with a high prevalence of malaria. And people who have this mutation in their hemoglobin, who do have sickle cell anemia, tend to have enhanced resistance to malaria. So we're going to end our discussion of all of this here. We'll come back and visit hemoglobin and myoglobin in future chapters. We'll come back and visit protein structure and function in future chapters as well. But this was a nice way to look at an actual protein, or two actual proteins in this case, and talk about how these are functioning and the binding within these.